Most enzymes can catalyze reactions in both directions, favoring the equilibrium concentration. In this way, the concentration of reactants and the concentration of products influence the activity of an enzyme. Other things affect enzyme productivity, but for now I want to just graph the effect of increasing temperature on an enzyme. Enzymes have evolved inside of very specific organisms who have lived in very specific environments. First, we'll look at temperature's effect on a human enzyme. The x-axis in the center graph will represent temperature, which will increase to the right. Reaction rate will be indicated by the y-axis, starting at 0 and increasing as it goes up. Humans are organisms that regulate their body temperature. So, inside of our cellular environment, the temperature stays around 37 degrees Celsius. As temperature increases, the productivity of the enzymes and therefore the reaction rate increases until the enzymes reach a critical point. This is the point at which the enzymes themselves denature because the molecular motion is too high. This means the hydrogen bonds break between backbone sections of the polypeptide and R groups of the polypeptide. The enzyme then falls apart. The temperature at which a thermophilic archaea's enzyme reaches maximum ability would look different. Despite the differences in temperature, the general graph would look the same. There would be a rise where maximum efficiency occurs, and then a sudden drop where the enzymes of the thermophilic archaea denature or fall apart. If we looked at a graph of enzyme-mediated reaction rate as a function of pH, we would see similar trends. There would be a pH at which the enzyme operates most efficiently. There will also be a pH above which the enzyme denatures or falls apart, as well as below which it denatures or falls apart. It would also look different depending on the environment that the cell we were talking about was functioning in. Now let's examine reaction rate in an enzyme-mediated reaction as a function of the concentration of substrate. At some point, every single enzyme is working on substrate, converting it to product as fast as it possibly can. It's at this point that the graph of reaction rate will even out. The reaction simply cannot go any faster. Enzyme saturation occurs when every active site on every enzyme is working as fast as it can on that specific substrate. At this point, there's only one way a cell can increase reaction rate. It has to make more enzyme. Other chemicals can mediate the ability of an enzyme to bind substrate and therefore turn it into products inside of a cell. The drawing on the left represents normal enzyme-substrate binding. The drawing in the middle represents competitive inhibition by another molecule that blocks the active site of the enzyme, preventing substrate from binding. The box on the right represents non-competitive inhibition. This is where an inhibitor chemical binds an allosteric site, forcing a change in the enzyme, not allowing the substrate to bind. Enzymes are a kind of protein that mediate chemical reactions in living things. They are biological catalysts. They increase reaction rate by greater than 1,000,000 times. The chemical that an enzyme affects is called a substrate. Enzymes are generally named after the substrate with the suffix ase, A-S-E, added to the end. So the enzyme substrate-ase would have a substrate called substrate. Or if an enzyme affected a chemical called dog, then the enzyme would be named dogase. The site where an enzyme binds its substrate is called the active site. Let's look at normal binding first. The enzyme binds the substrate. It lowers the activation energy required for the molecules to reach their transition state by providing a template, providing a microenvironment conducive to chemical reaction, temporarily bonding the substrate, and stressing the bonds in the substrate by stretching it. Sometimes an enzyme's activity is inhibited by other chemicals. First, let's look at competitive inhibition. Competitive inhibition occurs when an inhibitor binds the active site of the enzyme. This prevents the substrate from being able to bind the active site. Non-competitive or allosteric inhibition occurs when an inhibitor binds the allosteric site on the enzyme. This causes a shape change in the enzyme, inactivating it, preventing the active site of the enzyme from binding the substrate.